In 5.4.3, they want us to be able to state and also, to be honest, apply. I sometimes see a question like this on the IB exam where they're asking you to use this, apply this to find out uh, ancestry. So this is asking us to state that sequences different, um, or their differences actually accumulate. So they change over time slowly. So that allows us to identify who is more closely related to who. And not only that, because the way things change in our DNA, it's kind of predictable, like how much mutation occurs based on the protein. We can actually use it as a clock to identify how long ago did they diverge to form a common ancestor. So what I mean is that, and we'll draw our evolutionary tree again. We can use this tree to identify, hey, when did these species diverge? Was it 1 million years ago? Was it 2 million years ago? Was it half a billion years ago? And based on the number of changes and also what we know about the speed of changes, we can identify how long it occurred. Let's practice using this real quick. Just to to practice to see a question that they might ask you. So here's a good one. Which two species are most closely related? By using their sequence of DNA for a particular gene, perhaps. So we just pile these DNA strands on top of each other for our 1, 2, 3, 4 different species. So again, you can imagine them to be whatever species you want, maybe human, dog, chimp, bird, whatever you want them to be. And you just go across and identify differences. The first 4 nucleotides are the same, correct? However, when we get to the 5th nucleotide, we see a difference between these two species. Right here we have GG and here we have AA. Aha, here we have another difference, right? Yep, here we have a difference here. GGG looks good. AAAA, AAAA, GGGG, CCCCC, TTTTT, TTAA. So there's differences down here. TTTT, GGGG, CCGG. Here's 2 differences down here. The A's are the same, the T's are the same. Here we have a difference here. Thymine instead of guanine for those other ones. Cytosine, adenine. Okay, so those are the differences that we identified. Okay, so which 2 species are most closely related based on this image, based on this this information they gave us about DNA sequences? You should have noticed that species 3 and species 4, I didn't line up that arrow very well, those 2 are the most closely related. Why? Well, when we pile their DNA up on top of each other, the only difference is this one right here. So there's only one difference in their DNA, where the other ones have multiple differences. Between this one and species 1, there's 2 differences. Here, there's 6 differences, right? With this one, there is 5 differences. So yeah, species 3 and species 4 would be the most closely related. Not only can we use DNA to identify who's mostly closely related, but we can also use the amino acids they code for, right? Gene equals protein. We've said that multiple times. So your DNA will make your messenger RNA, and that'll eventually make the amino acids that will code for a certain protein. In this example here, we're looking at hemoglobin. Actually, we're looking at the beta chain, if you read that there. So hemoglobin is made of 2 alpha chains and 2 beta chains, and the sequence of those amino acids, those individual amino acids, will change over time, and we can use that to identify who's most closely related. So here, if we're comparing everything to the human, in this example, we don't always have to compare things to humans. But the rhesus monkey, there's 8 differences in its amino acid sequence in the beta chain. The dog has 32, The bird has 45, and this lamprey has 125 differences in its hemoglobin. So what does that mean? It means that humans and rhesus monkeys in this example are the most closely related. And we can do the evolutionary tree again. Here's human, here's monkey. I can draw another branch off here. Organism C, species C would be the dog. I can draw another branch off of here. Here's the bird. And I can draw another branch off of here. And here's our lamprey. So as we did with the DNA, right, we identified, I'm sorry, this is a mess. We can identify who's most closely related, but we can also, because mutations occur over time, those bases will change over time, we can also identify when they happened. And hemoglobin, you have a base change rate of 500 million years per base pair. Now there's lots of base pairs, right? So there's going to happen more of, but one base pair, about 500 million years for them to mutate. So we can identify when those changes in the sequences occurred to allow for our different amino acids. And this is called the molecular clock.